Hello students, I wanted to create an overview video on how to complete the cell homeostasis virtual lab. When you open your document, it'll look something like this. It's always a good idea to read through the document before you actually begin the lab. So I can see I'm going to use a link to access the lab. I'm going to follow the prompts and directions to get started with the lab. I'll start by making 5 different solutions. I'll follow all the directions to make different solutions, then I'll hit continue. I'll get the initial mass of 5 different bags, and I'll fill that in the data table below. Then I'll let 24 hours go by and record the final mass of each bag. I'll calculate 2 things, change in mass and percent change. And there's a chance to get some help with math if I need it. Then I'll look at this graph below and answer several questions about the lab. So that's what I will do. So let's get started. So here's the virtual lab. Let me start over so I can show you what it looks like. And we'll click Start to enter the virtual lab. Looks like to answer our question, we'll use different concentrations of dissolved sugar in a beaker to represent the cell's external environment. Then we'll use the same concentration of sugar in solution in dialysis, whatever that is, dialysis tubing to represent the cell. So here we go. I'll be taken into a lab space that has a variety of materials. At the top, I have some beakers. Looks like I have a graduated cylinder filled with water. I have some sugar. I have a stir rod. I have a petri dish, and I have a scale. So it starts by saying place beaker A on the table and fill it with 1,000 milliliters of water from the graduated cylinder. That one is my control group, so I can place it to this side. Now let's do the same with our experimental group. Here's B. Here's C. For C, I'll also be adding some sugar. So I put my petri dish onto the scale. I'm going to click this tare button so that the mass of the petri dish is ignored. Then I'm going to pour 50 grams of sugar into my petri dish. Awesome, looks like I got to 50. Now I'm going to take this, put it into the solution of 1,000 milliliters of water to get a 5% sugar solution. I can use the stir rod to mix it up real well. Now I'll make the rest of my solutions. All right, so now I have all of my beakers created with different percent sugar solutions. I have a control with no sugar, B with no sugar, C with 5% sugar, D with 10% sugar, and E with 15% sugar. Let's keep going. So we have some pre-filled dialysis tubes representing cells. So it turns out dialysis tubing is something that has a lot of similar properties to cells. So these aren't real cells, but they're models. One has been filled with water, and the rest have been filled with 50 to 100 milliliters of a 10% sugar solution. So now I need to get the mass of each one and record that data. So let's start with A. Looks like my mass here is 17.59. If I go over to my data table, awesome, 17.59. That one is already put in for me. Do the same for B. Looks like that one, oh, it's a lot less, 8.75. So let's go to my data table and write that in, 8.75. put it into the correct beaker, and keep going like that. Now I'll wait 24 virtual hours. All right, so now I need to see if there's been any change in the mass of my dialysis tubing, because that will tell me how much water has crossed the membrane represented by the dialysis tube. So let's start with beaker A. Seems like my final mass is 17.66. Let's put that into my data table. 17.66, already done for me. Awesome. Let's keep going. All right, now it's time to do some math. I need to create the difference between the initial and final mass of each one of these different dialysis bags. Then I also need to calculate the percent change in mass because they didn't all start with the same mass, so percent change will allow me to compare just a little bit easier. In order to do this, change in mass is just the final mass minus the initial. So I could take 10.40 and subtract 8.75. That would give me my change in mass. Then I can find the percent change in mass. And all I need to do for that is divide by the initial mass, divide my change by the initial, and then multiply by 100 so that it's a percent. If we need any help with this, then there's a spreadsheet that we can make a copy of. And if we make a copy of this particular spreadsheet, then here are the initial and final masses of each one of these. We can write them in this box, and it'll calculate change in mass and percent change in mass for us. Finally, I'll answer the questions. So down here, there's question A, B, C, D, E, and F. I'm going to write my answers right in this document. Then we're done. Please let your teachers know if you have any questions.